So I wanted to basically answer some questions. When do you use high throughput? When do you use SEC-SAXS? And very briefly, in a nutshell, if you're just going to do one sample, you're interested in one protein, in one conformation, and one condition, you definitely want to do SEC-SAXS. It is going to give you the purest results. You could run high throughput ahead of time. It's definitely the cheaper experiment. I definitely would recommend that if you can. But if you only got one sample, SEC-SAXS is the way to go. If you've got an engineering project and the people who've really been prolific in this is the Baker Lab. So for example, this is a slide I stole from my kids' engineering camp, where you want to first identify your goals, generate ideas, design, and then create, test, evaluate, and redesign, and then obviously publish. This is the sort of route you want to take. In structural biology, there's very few techniques where you can do this redesign cycle, where you can test multiple things over and over again and just try to figure out what's going on. And SAXS, a high throughput SAXS, is one of those things. So the example I give is the lock. We've been at this for centuries, a 6,000-year-old wooden lock from Egypt with a lock and key with a wooden key. These are Roman keys. If you look at your current key setup and lock, it's a very complicated thing. It's got springs, it's got screws in places that you would never think and understand. And these are a lot like our proteins. We have a lot of amino acids. We don't understand why they're there. We have lots of parts that we might want to add to and see if it works better. There are very few techniques that allow you to tinker. So here's lysozyme, for example. We just don't have this ability with a lot of techniques to go through and test each of these amino acids and modify our hemes and things like that. But you can do that with SAXS. So, these are famous, proteins, and DNA are incredibly versatile in terms of creating structures. And, here's cryo-EM versions of what people tried to design. Here's just a simple helix turn helix system by the Baker Lab. And here are all the different topologies that computationally might be able to be set up just using the helix turn helix motif. And so we have gone through all of those constructs for the Baker Lab and identified, here's the example, which ones are real and which ones are not makeable. And so they ran all of these, I think it's something 44 constructs and more of just helix turn helix. And they did all the SAXS, all the P of R functions, they did some CD, they ran their gels, etc. And they did that for all 44. And so basically these are the sort of techniques that you can use for such high throughput analysis. So you would never want to run, you are unlikely to get the time or the capability to run all of these in SEC-SAXS at once. And especially if you want to do like buffer conditions or things like that, it's just not going to happen with SEC. And it's something that we can do with high throughput. So this is the beamline that we've been working with, what we've done. And I've showed this a lot. So for those of you who've never collected data at SIBYLS, we try to do it every week or every 2 weeks where we have a smaller workshop, an hour-long workshop on processing the data from that week. And during that time, I'll try to highlight, the issues that came up during the problem, and I'll try to be much more specific about the examples. So if you've never collected data or have collected that data that week and just want to go over something, we do hold these sort of hour-long office hours once every 2 weeks, once every week, to go along with your data collection. So we're really trying to push the limits of this batch mode high throughput data collection. And again, the people who really have benefited from this are the protein engineering systems like the Baker Lab. I mean, they're publishing Science and Nature papers left and right. And I'm hoping that at least their grad students or other people will take notice and start doing the same. Right now, we're 2 hours per 96 well place, 30 microliters. We've basically designed a system where you can shoot the X-ray beam through the head of our liquid handling needle, and there's all kinds of advantages for it, for doing that. And I will put it in slideshow mode. Current slide, sorry. Let's see. You probably can't see the movies, so I won't even bother. So this is a data collection on multiple data collections on GFP. If you can see the movie going, you can see it changing from clear to green. And these are repeat buffer sample collections. And of course, I happen to choose an aggregated version of this for this example, but that's what I made the movie from. All right, moving on. So it's important to know how much concentration. Everybody asks, like, what is the concentration I'm requiring? Here is a very simple rule of thumb. You take your molecular weight, you take 100 and divide your molecular weight through that, and it should be greater than the mgs per mil you'll need. Okay, so if you have 100 kilodalton protein, about 1 mg per mil, 200 kilodalton, half mg per mil, etc., or 50 kilodalton, 2 mg per mil. Those are kinds of rule of thumb to apply. All the different other techniques, so I've got it listed out here, sort of the requirements. Mikhail will talk more about the SEC, requirements. But again, it's all about your signal to noise and what you really want to do with your sample. So if you just want the shape of everything, you use that rule of thumb I just mentioned, sort of 100 KD. All you really need is high quality data up to a Q of 0.15, and it will give you a pretty reasonable shape estimate. If you are looking for conformational flexibility or contrasting things that are higher resolution than just the shape, and that the conformational changes might be smaller, you'll need to probably go 3 times that concentration, because you really want high quality signal beyond 0.15, where the data is enriched in the small conformational changes, the small data. So again, it's reciprocal. For the low resolution information is highly weighted in the small q regime, that is the longer distances are highly enriched in the small dist angle data, and then the more subtle conformational changes, fold stuff, is in the higher q. Although, of course, everything contributes to everything, but that's sort of an enrichment. If you want shape, you want purity. If you want conformational flexibility, you want purity. But if you just want to see if mutations or ligands are changing things, you don't have to be perfectly pure. You can take something that's slightly aggregated and start adding ATP or some other thing to it, and you might see conformational changes for it. So the purity is less important, concentration is important, and again, you're just modifying one protein example over and over again. So just things to think about. So we've been going through these needle processes. You can see some legacy needles in the bottom here, and actually our newest needle is this guy right here. You guys probably can't see the movie, but basically,
you see.
Okay, the needle is 3D printed these days, and we've been able to protect the mica window as it enters a covered plate by recessing the window, et cetera, et cetera. So there's been a lot of iterations of this, and we've arrived at this one. And again, we're always iterating beyond this to both improve our success rates and decrease the volumes required. Let's see. So what are our overall goals? It's to provide the highest quality data to the largest user community we can. We want to minimize what you need to send us. And we want to provide a novel platform for screening variants, conditions, and metabolites. And then also a side effect, or actually a pretty important effect for increasing our speed, is that we make time for other techniques, development of other techniques like SEC or like time resolve stuff, which we have yet to implement at SIBYLS. So here's an example protein screen where we're screening salt and pH. So you could imagine setting up where the yellow things are buffers. There's 2 unique buffers for every sample. And we're stepping through the first number is the pH. The second number is the salts. So you can imagine taking one protein and screening all of these conditions for 30 conditions on a plate. And we've done that several times. This would be an example of a construct screen where you have a buffer and 3 concentrations of the same protein construct, moving on to the second construct, the third construct, etc. So 3 concentrations for that. So Catherine's been through this with you guys. She's shown you how to click time and access the sample. The hope is that from the conception of your experiment to collection, it should be like 2 weeks. So we're trying to really make SAXS data collection available to all very quickly in High-Throughput. She's gone through that. All right, some of the things to be aware of with the data. And this was a question from a, you know, what happens here. So, you know, Jesse mentioned that there's radiation damage as you expose your sample, and you can never tell ahead of time when that will occur for any given sample. So when we place your sample in front of the beam, as I showed on those needles, we are going to collect about 30 to 50 frames, depending on radiation damage, exposure, etc. You're going to receive all those 33 frames. In the best case scenario, there's no radiation damage, and you can just average all of those 10 frames together. And what I'm showing here is the timeline here, 10 seconds, and then the detectors integrating over this 0.3 seconds, and then it dumps the data to a file, then it integrates 0.3 seconds, dumps it to a file, etc. So you end up with 33 pictures of the scattering that you put together. And you have to decide when to cut it, okay? So here are the packages you might receive. This is a directory tree that you'll get from us, or Catherine will email you, or someone will say, hey, your data has been collected and we've made it available. And so you'll see in your directory tree these folders, generally. There's going to be something called an average graph. I'll show you this. Some radius of gyration analysis, the sample cell picks, and then your subtracted data. Now, because you sent us buffer before and buffer after your sample, in the subtracted data, you will not have any of the buffers there. There will be no analysis in the subtracted files for that. What you will have per well that had sample in it, you'll have that buffer, that first buffer subtracted from your sample, and the second buffer subtracted from your sample, and then the average of the 2. And so there'll be 33 frames in this directory. You also have access to the unsubtracted data, okay, which is Rao's program can use, and you can do a manual subtraction with that data. And in that case, you do have analysis of your buffers, right? So buffer 1 and buffer 3, they're there, but not in the subtracted data. So I'm going to step through. These are things that I showed in my weekly sessions, so I will not go through them. So I will go through a little bit in detail. So that avgraphs folder, which is the first one that was on that list, you'll be able to see basically a summary. And again, depending on what operating system you have, you can display it in this way, right? And of course, this is super low resolution. Sorry about that. But generally, you can put it in some kind of view mode where you can look at all of the images immediately and assess which wells are worth analyzing, kind of get a sense of your whole data. Again, I don't know if this movie will play for you, but this is an example of the average graphs. It'll probably click slower through for you than it is for me. And these are comparing buffer 1 and buffer 2 to one another, how the subtraction went. Occasionally, you'll see in the high Q region that there's some disagreement, like, or in the low Q region that there's some disagreement. But that gives you some information that you're going to have some trouble in that particular sample. You'll be able to see how much radiation damage there is. So for every particular image, we've integrated the first 4 frames, comparing buffer 1 and buffer 2, or first 5, and then all of them together. So if there's a difference between the first 5 averaged together and all of them averaged together, that means radiation damage has occurred somewhere during that sample. All right, so let's see, moving on. Here are the sample cell pics, and again, I just have it too low resolution for you to see the details, but somewhere over here, even in this low resolution view, you can see that there's a bubble in D2, for example. So you can go through the sample cell pics and quickly assess where bubbles occurred, and look at the average graphs and also see maybe that bubble, you were lucky and that bubble only occurred in one of your buffers but not the other buffer, right? All right, so then here's the extracted RG region. So in this extracted RG region folder, there is some guidance on when radiation damage happens. There's an HTML file which should be able to open via a browser of some kind. And in it, it gives you an estimate of the radius of gyration and the number of files that the program thinks you can use to preserve that same radiation of radius of gyration. It also gives you an estimate of sort of the number of points that are in that region. And it tells you whether the buffers agreed in the low Q region, whether the sample is radiation sensitive, whether it was an aggregate, some judgments. Also in this extracted RG region is all of the RGs of your curves. So all the DAT files, these are all truncated RG regions of each of your curves that were generated there. Where the Guinier region was flat. So this data can be directly put into a publication. Sometimes you'll see, you know, as part of the scattering profile, someone will put in as a subplot in the top corner, the Guinier region and how it fits the data. So this would be the data you could put in that figure. So that's all there. So then,